The title of the project is Glucose-conjugated Camptothecin loaded glutenin nanoparticles for treating breast cancer. IMR Roger Rajeshwari work under the guidance of Dr. K. Selvaraj, Assistant Professor in Department of Biotechnology, Kalasalangam Academy of Research and Education. Introduction. Breast cancer is the leading cause of death in women compared to other cancers. Treating breast cancer is the greatest challenge due to altering genetic and environmental factors. The drug provided to the body is carried to any tissue with the help of plasma cells. Conventional chemotherapy treatment for cancer has shown many side effects. Conventional drugs not only target cancer cells but also disturb normal healthy cells and leads to drug resistance against cancer cells. Cancer cells are rapidly proliferating. Therefore there is an overexpression of receptors, thus differing from normal cells. Before making a drug for breast cancer treatment, one has to consider the drug's solubility, selectivity, and the biocompatibility of the drug. There are strategies to enhance the delivery of drugs to the cancer cell by receptor-mediated drug delivery, carrier-mediated drug delivery, and glucose can enhance the circulation of drugs in the body. According to the need, we should design a drug with help of these strategies. We make use of GLUT1, glucose transporter receptors, as target, and proteins glutenin as a carrier. Here the image on the left represents the drug formulation. The drug was encapsulated into glutenin protein. The carrier glutenin was conjugated with glucose. The conjugated glucose selectively delivers the drugs to the GLUT1 receptor. Through the GLUT1 receptor, the drug enters the cancer cell and leads to apoptosis by interrupting mitochondria and nucleus. Glutenin extraction from wheat flour. First, the wheat flour was made into dough. The dough was washed in running water to remove starch from it. Then soaked in ethanol for 1 day. Glutenin obtained was dried and grinded to obtain smaller particle polysaccharide synthesis. 2 grams N-succinyl glucosamine, 2 grams sodium methoxide, 1 gram succinic anhydride, 3 milliliters triethylamine was dissolved in 40 milliliters ethanol. Then keep the solution in magnetic stirrer at room temperature for 12 hours. Polymer synthesis. 1 gram glutenin was dissolved in 50 milliliters ethanol. And then 0.9 grams of EDC, dimethylamino-propyl 1,3-ethylcarbodiamide, and glutaraldehyde were added to it. This was kept under pressure for 48 hours. Drug encapsulation and conjugation. For drug encapsulation, half a microliter of the polymer was dissolved in 50 milliliters of water, and 2 milligrams of camptothecin mixed in 50 milliliters ethanol was added to it. 2 drops of glutaraldehyde were added. The prepared solution was kept for 12 hours in stirring. For conjugating glucose, EDC, dimethylamino-propyl 1,3-ethylcarbodiamide, and 1 mg polysaccharide prepared was added and kept in magnetic stirrer for 12 hours. This was the image of the final product. In our work, we have synthesized a glucose-conjugated camptothecin-loaded gluten nanoparticle formulation which helps in overcoming the disadvantages of conventional drugs. The stability and absorption efficiency of the drug formulation was high, which was predicted by drug stability study and drug dissolution profiling. Through kinetic models the mode of drug dissolution was interpreted. Through the cancer cell line study against MCF7, breast cancer cell line, it is proven that the drug formulation has a cytotoxic effect.